Welcome back. Today, we're zooming in to explore the microscopic anatomy and organization of skeletal muscle. Let's get started.
Let's zoom in past the visible muscle to the cellular level. A single skeletal muscle cell is known as a muscle fiber. Surrounding this fiber is the sarcolemma, or the plasma membrane. Because these cells are so large and active, you'll notice many nuclei tucked just beneath the sarcolemma to manage the cell's functions.
Inside the fiber, the space is packed with myofibrils, long cylindrical bundles of protein. These are made of even smaller myofilaments, the actual machinery of contraction. We categorize these into two types. Actin, thin filaments. These are guarded by two regulatory proteins. Tropomyosin winds around the actin, covering the binding sites. Attached to it is troponin, a globular protein that acts like a lock. When calcium binds to troponin, it pulls tropomyosin away, exposing the actin so contraction can begin. Myosin, thick filaments. These feature myosin heads, which act like small oars that reach out to bind with actin. The functional unit, the sarcomere. The myofibril is organized into repeating units called sarcomeres, the basic functional unit of contraction. Z-lines mark the boundaries and anchor the actin. The A-band is the dark central region where thick myosin filaments reside. The I-band is the lighter region at the ends, containing only thin actin filaments. To trigger a contraction, the signal must reach deep into the cell. T-tubules are invaginations of the sarcolemma that carry the action potential into the interior. These tubules run alongside the Sarcoplasmic Reticulum, a specialized network that stores calcium. The enlarged ends of the reticulum are called Terminal Cisternae. Where a T-tubule is sandwiched between two terminal cisternae, we find a triad, the critical junction where electrical signals are converted into the chemical release of calcium.
A muscle doesn't move on its own. It needs a signal from the brain. The site where a somatic motor neuron meets a skeletal muscle fiber is called the neuromuscular junction, or the myoneural junction. Think of this as the handshake between the nervous system and the muscular system. The signal doesn't just hit the muscle anywhere. It targets a very specific region of the sarcolemma called the motor end plate. This is a specialized area of the muscle fiber's membrane that is packed with receptors, specifically designed to receive the chemical message sent by the motor neuron's synaptic terminal.
While one neuron connects to a specific spot on a fiber, that single neuron usually controls more than one fiber at a time. This functional group, consisting of one motor neuron and all the muscle fibers it innervates, is called a motor unit. In muscles requiring precision, like those in your eyes, a motor unit might control only a few fibers. In large power muscles, like your quadriceps, a single motor unit might control hundreds of fibers at once, ensuring they all contract in unison.
The protective layers. Skeletal muscles aren't just bundles of cells. They are highly organized by layers of connective tissue that provide structure and support for nerves and blood vessels. We can think of these as protective wrappers at three different levels. Endomysium, the inner layer. At the most microscopic level, each individual muscle fiber is wrapped in a delicate layer called the endomysium. This ensures that every single cell is insulated and anchored to its neighbors. Perimysium, bundling the fascicles. Moving one step up, muscle fibers are gathered together into groups called fascicles. Each of these bundles is surrounded by a thicker layer of connective tissue known as the perimysium. When you look at a piece of meat and see the grain, you are actually seeing these fascicles. Epimysium, the outer shell. Finally, the epimysium is the tough outermost layer that surrounds the entire muscle. It binds all the fascicles together and blends into the tendons that anchor your muscles to your bones, allowing them to pull as one cohesive unit.
Now, let's look at the second type of muscle, smooth muscle. Unlike the long, multi-nucleated fibers of skeletal muscle, smooth muscle fibers are spindle-shaped and much simpler in their microscopic design. Each cell contains only one nucleus, which is centrally located within the fiber. Involuntary control. varicosities. Smooth muscle is under involuntary control, meaning it is managed by the autonomic nervous system. Instead of a single motor end plate, the autonomic nerve endings spread out across the muscle fibers. These nerves have swollen regions called varicosities. These varicosities act like sprinklers, releasing neurotransmitters over a wide area to signal the smooth muscle fibers to contract.
Finally, let's examine cardiac muscle, found exclusively in the heart. Like smooth muscle, these fibers are involuntary and typically contain only one centrally located nucleus. However, you'll notice they are branched and striated, designed for the high-pressure demands of pumping blood. The power of intercalated discs. The most defining feature of cardiac muscle is the intercalated disc. These are specialized junctions where the ends of individual cardiac cells lock together. They serve a dual purpose: structural support. They physically anchor the cells together so the heart doesn't pull itself apart during a powerful contraction. Electrical communication. They allow electrical signals to pass almost instantly from one cell to the next, ensuring the entire heart muscle contracts as a single coordinated unit. Autonomic regulation. Similar to smooth muscle, the heart is regulated by autonomic nerve endings. While the heart has its own internal pacemaker, these autonomic nerves adjust the heart rate and strength of contraction to meet the body's changing needs.
Thank you for watching. I hope you learned something new today. Please don't forget to like and subscribe to stay informed for our next lesson.